In Chapter 10, we're going to look at vision and how we perceive vision. And we'll deal with these topics here. We'll start off right now with what light is and how the eye works. To talk about vision, we need to talk about what the stimulus is. In hearing, the stimulus was sound waves. In vision, we're also dealing with waves, but these are light waves. They are an example of electromagnetic energy. And there's a lot of different types of electromagnetic energy. There are gamma rays, X-rays, radio waves, and so on. All of those are electromagnetic energy that travel through the air in waves just like light does. The only difference is that what we see are wavelengths of between 400 and roughly 700 nanometers in wavelength. And so to get us thinking a little bit about what these waves are, here's a short video.
Normally, we see light waves that bounce off objects. They beam into our eyes and onto our retina at the back of our eye, where an upside-down and backward image appears. It's turned into electrical impulses that race to the brain. Which allows us to see what we need to survive. But there's a lot we miss. We only see the rainbow of light waves called visible light. But that's just a fraction of the millions of wavelengths in the vast electromagnetic spectrum.
The visible spectrum that we see is classified according to wavelength, which is the distance from one wave to the next. Light between 400 and 700 nanometers in wavelength is able to be detected by the receptors at the back of the eye here in the retina. So let's take a look at the eye and its critical role in helping us see. Light from the sun or from any artificial light, any light source, travels through the air in the form of light waves and will be absorbed and or reflected by an object. So most objects are going to reflect some light. That light then again continues traveling off the object, enters the eye through an opening called the pupil. The pupil is the dark spot in the center of a person's eye, and it can get larger in a dark environment to help us see better, and it can get smaller in a very bright environment to keep a optimal level of light for us. So the light enters through the pupil. It gets focused by the lens and ideally winds up at the back of the eye here in the retina in a sharp image. We have other aspects here of the eye that we should look at. The outer covering of the eye is the cornea. That is, in a healthy eye, clear so that most, if not all, of the light wave can pass through it. The iris is the colored part of the eye that we see. The lens, notice, is attached by these ciliary muscles. These muscles control the lens. And if you take a moment and look at your thumb and bring it up really close to your eyes, you might get those ciliary muscles to work a little bit. They're going to squish the lens a little bit and help you focus on something that would otherwise be too close to be able to see clearly. So they play an important role. In any case, the light that travels to the back of the eye is absorbed by the receptors in the retina. And notice there's an area here of the retina called the fovea. The fovea is the point of central focus. It's where an object that we are looking directly at gets processed. And it's an area that has a high density of visual receptors, allowing us to see in the greatest amount of detail. All the axons from the cells in the retina leave the eye and form the optic nerve. Where they poke through the eye, there are no receptors, and that creates a small blind spot that you may have heard of. We don't normally notice the blind spot because our eyes are always in motion and our brain fills it in, but there are ways that you can detect it.
In principle, the working of the eye is similar to that of the ordinary camera.
When you look at human vision and compare it to insect vision, it's quite difficult to compare them. When you compare them, you really start to appreciate what makes human vision unique.
The lens of the eye forms an image on the sensitive surface of the retina.
The human eye is called a camera eye because the retina is like the film in an old camera. And this is where the lens focuses the light.
Have a look at your eyes. And when I start to look at the back of the eye, I can see the retina.
Magnified many hundred times, the retina is seen to consist of this complicated arrangement of rods and cones.
The rods are really sensitive to light and help us see at night, whereas the cones are adapted to see detail and color.
So as we saw in the video, the retina includes two types of receptors, rods and cones. The cones help us see detail and color, but they require a large amount of light to work, so they are not really able to do much for us in dim light. The rods are the receptors that work best in dim light, but they are not good at helping us see detail and color, but they are what allows us to get around when there isn't that much light available. Now in the retina, the last layer of cells are the rods and cones, and here where you can see these discs shown here in purple in both rods and cones, that's where there is a visual pigment molecule that absorbs the light. And the job of a rod and cone is to convert that light energy into a neural signal. That's why they're receptors. They convert light energy into a neural signal. And they in turn are connected to this next layer of cells called bipolar cells. And then finally to the ganglion cells. The axons of the ganglion cells leave the eye and send that information to the brain. They form the optic nerve. In addition to these three layers of cells, the receptors, the bipolar cells, and the ganglion cells, you can see that there are these additional neurons that run laterally. There's a first layer between the receptors and the bipolar cells called horizontal cells. And then another layer here, right before the ganglion cells, called amacrine cells. And they help with creating additional contrast for our vision. So here's a summary of the rod and cone system. The rod system functions best in dim light, poorly or not at all in bright light. Detail vision is poor, does not help us with color. They are found mostly in the periphery of the retina, not in the center of your field of vision. Their receptive fields are large. That means each individual rod, the way it works is it's going to have a larger chunk of the world that it sees. And in addition, the ganglion cells that receive information from rods are going to gather information from many rods. And so the information that the brain gets is really from a very large area of the visual field, which is why the rod system is not good at seeing detail. It doesn't have pinpoint accuracy, but it sees the big picture. And because it sees the big picture, it can bring in light from a very large chunk of the visual field, allowing them to work even in dim light because they kind of gather all that light together. The cone system, you can see here, works differently. They work best in bright light, not at all or very poorly in dim light. Detail is good. Color vision is possible. They help us distinguish between colors. They are found mostly in the center, in the fovea, where our highest resolution vision is, which makes sense. They have small receptive fields, and in addition, only one or a very small number of cones will come together onto a single ganglion cell, allowing the brain to receive a very high resolution, high detail image of what is being seen in the visual field. So how do these rods and cones signal the presence of light? We actually are going to see that they are working in a way that's sort of opposite of what we would think. In the dark, the rods and cones are continuously active. They are a neuron. They are releasing the neurotransmitter glutamate, and basically in the dark, that glutamate is constantly being released. And so the default is a constant release of glutamate. And that works because there's this a sodium channel that's gated through something called cyclic GMP. Don't worry too much about that, but do know that the sodium channel is being kept open, being kept in this depolarized state all the time. Now, they don't produce action potentials. They produce what are called graded potentials, but they're still releasing this steady stream of glutamate. And the bipolar cell that might be connected to it is either depolarized or hyperpolarized. So it can either become excited or inhibited, has that flexibility. In contrast, when we turn on the light and the light gets absorbed by the rhodopsin, the visual pigment molecule inside here of the rod in this example, that closes the sodium channels. With the sodium channels closed, there's no more excitation, no release of glutamate, and then the bipolar cell goes into the opposite state. If it was being depolarized in the dark, it'll be hyperpolarized now. If it was hyperpolarized in the dark, it'll be depolarized now. So it's like a switch. You're flipping a switch on or off. The only thing that's a little unusual is that this rod or cone is in a constant state of activity in the dark. And we normally think of we would think that the receptor would be turned on by light, but actually its default position is to be on, and it gets turned off by light, and the reaction here gets turned off when the light turns on. The bipolar cells stimulate the ganglion cell in a way that's proportional to the intensity of the light. And again, there are no action potentials here. There's also, to help us gain more contrast, we have the horizontal and the amacrine cells. These are the guys that run laterally. And as an example, a horizontal cell, if there's light coming down here, it'll reach around and it'll inhibit the neurons next to it. Why? Well, that'll create an extra big difference between where there's light and where there's dark. Really high contrast, allowing us to see better. The ganglion cells that gather all this input, they will receive information from either rods or cones, but not both, and then they send it to the brain.